Skeletal muscle damage and regeneration are principal features of the physiological adaptation to exercise 28, 46. Acute bouts of moderate to vigorous exercise can induce structural damage in muscle fibers 46, 47, which triggers an initial intramuscular pro-inflammatory immune response characterized by neutrophilia 48, 49, CD8 plus T cell accumulation 50 and increased numbers of monocytes and macrophages, the latter of which are functionally shifted towards a pro-inflammatory M1 phenotype, classically activated, 51, 52, Fig. 2b. This process is mediated by pro-inflammatory cytokines released from damaged muscle tissue, including tumor necrosis factor, TNF, encoded by TNFA, CC-chemokine ligand 2, CCL2, MCP1, and interleukin-1 beta, Illinois 1 beta, 49, 53. Resolution of inflammation following exercise-induced muscle damage is less defined. In models of muscle damage and repair, for example, cardiotoxin, notoxin, hind limb unloading and/or reloading, inflammatory resolution is associated with a phenotypic shift of macrophage populations towards an anti-inflammatory, reparative M2 phenotype, alternatively activated, 54. Many factors mediate this transition from M1 to M2, including macrophage and regulatory T, TREG, cell-induced interleukin-10, Illinois 10, 51, 55, macrophage-induced insulin-like growth factor 1, IGF-1, 56 and phagocytosis of cellular debris by M1 macrophages 51. Exercise-induced muscle damage is postulated to closely mimic this response. However, direct comparisons are likely imprudent given differences in the degree of damage that occurs between experimental techniques and exercise 53. Of note, the use of the M1, M2 macrophage polarization paradigm is likely oversimplistic, as macrophages can adopt a broad range of multidimensional phenotypes in response to various stimuli 57, including exercise 51, 58, 59, as discussed in more detail below. Exercise is a potent whole organismal environmental challenge that perturbs numerous physiological processes that are regulated by several integrated homeostatic control circuits that operate at the cellular, tissue and systemic levels. A detailed overview of the design and regulation of homeostatic systems and disease susceptibility is provided by a recent excellent review. Emerging work demonstrates that homeostatic control circuits are maintained by inter-organ signaling. Such signaling is achieved by the secretion of hormones, cytokines and growth factors into the host systemic milieu from various tissues and organs, for example, skeletal muscle, heart, bone and adipose tissue, which can subsequently regulate diverse processes in local or distant organs via paracrine and endocrine regulatory feedback loops. Over time, we speculate that chronic exercise-induced perturbation of inter-organ signaling promotes physiological adaptation across homeostatic control circuits, establishment of a higher homeostatic set point in concert. Such adaptations stimulate reprogramming of the systemic milieu, potentially characterized by alterations in the availability, reservoir, mobilization, recruitment, retention and function of specific cell types and/or molecules. In turn, this reprogramming of the systemic milieu might consequently alter the nature and strength of instructive signals at the tissue and cellular levels in the environment of distant organs that potentially harbor transformed cells or a pool of cells primed for malignant transformation. Fig. 3. Surprisingly, little is known about the mechanistic effects of exercise on tissues that are distant from those directly implicated in the exercise response. Consequently, there is a paucity of data on how exercise modulates the function, biology and programming of cells that reside within distant tissues, organs or cellular compartments. In this section, we speculate on how exercise might reprogram distant tissues harboring tumor cells, that is, the TME, focusing on three key processes that are known to be regulated by exercise in skeletal muscle and are critical components of the TME that influence cancer hallmarks, metabolism, angiogenesis and immune response. The pleiotropic nature of exercise suggests it might plausibly influence the entire breadth of TME contributions to cancer hallmark capabilities. However, this would be highly conjectural given the current lack of available data and, therefore, will not be discussed. In non-transformed cells, nutrient uptake is strictly regulated in a non-cell autonomous manner by growth factor signaling or extracellular stimuli. For example, PI3K is activated by growth factors, for example, insulin and IGF-1, in the tissue microenvironment. This places a high degree of reliance on sufficient mitogenic growth factor availability to sustain proliferation in transformed cells. As such, in cancer cells, metabolic signaling pathways downstream of growth factor receptors can be constitutively activated, permitting an adequate uptake of nucleotides, proteins and lipids to support the bioenergetic and biosynthetic demands of hyperproliferation. Metabolic reprogramming, most predominantly the switch to aerobic glycolysis, is considered a key intrinsic feature of cancer cells but one that remains subject to modulation from the TME. Nutrient and growth factor signaling. Prolonged physical inactivity results in excess nutrient and growth factor availability in the systemic milieu and presumably in the TME. Growth factors such as insulin and IGF signal through their respective receptor tyrosine kinases, which activate the major signal transduction pathways, for example, the PI3K ACT, MAPK and MYC pathways, stimulating the uptake of nutrients to support proliferation. By contrast, chronic exercise reduces growth factor availability in the systemic milieu, subsequently improving and/or restoring whole organismal metabolic homeostasis. For example, chronic exercise substantially increases glucose uptake and clearance in skeletal muscle via insulin-independent mechanisms, leading to decreased basal levels of insulin, IGF-1 and glucose in the circulation. Randomized trials indicate that chronic exercise might confer similar favorable effects in patients with primary cancer. Although overly simplistic, exercise-induced restoration and/or maintenance of whole organismal metabolic control might, in turn, alter homeostatic control signals that regulate nutrient and growth factor uptake and/or availability at the tissue level and ultimately cancer cell level, causing downstream effects on metabolism and bioenergetics. Data to support this notion are emerging. In a chemically induced model of mammary carcinogenesis, physical activity, wheel running, caused a delay in tumorigenesis, with concurrent reductions in circulating levels of insulin, IGF-1 and leptin. These reductions at the systemic level occurred in conjunction with alterations in intratumoral metabolic signaling, signified by increased activation of AMPK and reductions in levels of activated AKT and mTOR. Similarly, in a TPA-induced SENCAR, sensitivity to carcinogenesis, mouse model of skin cancer, 10 weeks of forced treadmill running reduced the activation of PI3K, AKT and ERK1/2 in skin tissues. In 3 distinct, Claudin-Low, triple-negative syngeneic breast cancer mouse models, forced treadmill running either inhibited, had no effect or accelerated tumor growth compared with the sham control. Metabolomic profiling of excised bulk tumors indicated differential alterations in tumor metabolic programming between models, although whether these differences were responsible for the differential tumor response to exercise was not determined. Interestingly, a series of observational studies suggest that the association between exercise and disease outcomes in patients with colorectal cancer, CRC, might differ on the basis of intratumoral expression of factors involved in cellular metabolic regulation. For example, in tumors negative for the activation of WNT signaling, through the nuclear accumulation of beta-catenin, also known as CTNNB1, post-diagnosis exercise was associated with a marked reduction in cancer-specific mortality. The protective influence of exercise was not observed in patients with nuclear cTNNB1 positive tumors. WNT signaling has a critical role in many biological functions in cancer, including metabolism. In subsequent work, exercise was associated with substantial reductions in CRC-specific mortality in patients with tumors with negative or low expression of insulin receptor substrate 1, IRS1, whereas patients with tumors with high IRS1 expression did not appear to benefit from exercise. IRS1 is a cytoplasmic substrate of the insulin receptor, INSR, and IGF-1 receptor, IFG1R, signaling pathways and is aberrantly activated in numerous cancers. Whether the antitumor effect of exercise is altered by the activation status of specific tumor metabolic signaling pathways has not been investigated in preclinical studies. However, there is indirect evidence from caloric restriction models to support this hypothesis. For instance, human xenograft tumors in mice with activating mutations in PIK3CA and/or PTEN loss are resistant to the anti-cancer effects of caloric restriction, despite systemic reductions in insulin and IGF-1. However, restoration of PI3K signaling in cancer cells resensitized xenografted tumors to the anti-cancer effects of caloric restriction. Autophagy, another process that might contribute to exercise-dependent regulation of tumor metabolism, is autophagy, a process by which cellular components are degraded to maintain cell viability and homeostasis in response to stressful conditions. When extracellular nutrients within the TME are depleted, cancer cells utilize autophagy to support growth and proliferation, suggesting that inhibition of autophagy might be a viable anti-cancer approach. However, context is critical, as autophagy can be tumor suppressive, for example, via elimination of damaged organelles, as well as tumor promoting, in established cancers, for example, via intracellular recycling to provide essential substrates for metabolism. In the context of chronic exercise, a transgenic mouse model with normal autophagy under basal conditions but a deficiency in stress, exercise or starvation, induced autophagy in skeletal muscle had decreased exercise endurance and altered glucose metabolism following acute and chronic exercise. In addition to effects in skeletal muscle, exercise also induced autophagy in the liver, pancreas, adipose tissue and cerebral cortex in this transgenic mouse model. Whether exercise-induced stress activates autophagy in tissues that harbor malignant cells, or cells primed for malignant transformation, or cancer cells themselves within those TMEs, and whether such effects inhibit or potentiate tumorigenesis, is not known. Lactate, the metabolic status or programming of cancer cells, also alters the metabolic composition of the extracellular milieu, influencing the fate of other genetically stable cells in the TME. One important manner in which cancer cells might exert this effect is via the accumulation of lactate as a result of the high utilization of extracellular glucose and glutamine. Increased levels of lactate in the TME cause a number of pro-tumorigenic alterations, including promotion of an immunosuppressive environment via immune cell recruitment and/or reprogramming, tumor cell invasion and angiogenesis. In a mouse model of breast cancer, using the human MC4L2 cell line, 7 weeks of forced treadmill exercise inhibited tumor growth and reduced lactate concentrations in bulk tumor lysates, with concomitant reductions in monocarboxylate transporter 1, MCT1. Although not investigated, it could be speculated that reductions in lactate concentrations are the consequence of either exercise-induced reductions in nutrient, glucose, availability, which stimulates cancer cells to shift from glycolysis as the predominant form of ATP production to alternative processes that generate lower levels of lactate byproduct, for example, oxidative phosphorylation, or enhanced tumor vascularization following acute or chronic exercise exposure, which enhances the clearance of extracellular lactate. Solid tumors have abnormal blood vessels, leading to impaired oxygen delivery and low oxygen tension, hypoxia. Hypoxia activates HIF-1, which regulates a highly conserved cellular response mechanism that involves a wide variety of critical cellular processes that are hijacked by tumors, including invasion, metabolism and angiogenesis. Unlike physiological angiogenesis, HIF-1 induced angiogenesis in solid tumors paradoxically causes pathological angiogenesis, characterized by abnormal blood vessel formation and impaired perfusion and oxygenation of the tumor, thus creating a self-perpetrating, vicious cycle of hypoxia and a more aggressive tumor phenotype. Given the aforementioned regulatory effects of exercise on physiological angiogenesis in skeletal muscle, investigation of whether such effects translate to solid tumors has received attention. In a syngeneic mouse model of breast cancer, 4T1 cell line, voluntary wheel running, that is, in cage wheels permitting ad libitum running 24 hours per day, increased the density and maturity of the tumor vasculature, as assessed by the presence of CD31 vessels and desmin-positive parasite coverage of capillaries, respectively, which occurred in concert with reduced intratumoral hypoxia and inhibition of tumor growth relative to sedentary mice. Several additional studies have corroborated this finding in syngeneic mouse models and xenograft models of breast and prostate cancer. Several mechanisms might underpin these effects. First, exercise increases vascular maturity in solid tumors. Through enhanced association between desmin-positive, chondroitin sulfate proteoglycan, NG2, positive parasites and CD31 endothelial cells. However, this phenotype occurred in concert with reduced expression of PDGFRB, which encodes platelet-derived growth factor receptor beta, PDGFR-beta, in bulk tumor samples, suggesting that alternative mechanisms might be responsible. Second, exercise increases shear stress on the vascular wall, leading to upregulation of angiogenic factors, for example, VEGF-A, osteopontin and macrophage-inflammatory protein 1-alpha, MIP1-alpha, within endothelial cells, which in turn induce transcriptional activation of nuclear factor of activated T cells, NFAT, via nuclear localization of NFAT cytoplasmic 1, NFATC1, and subsequent expression of thrombospondin 1 within endothelial cells, thus stimulating angiogenesis. Finally, exercise increases the bioavailability of nitric oxide, a critical mediator of tumor angiogenesis and metastasis, vascular maturity and lymphatic vessel function. An acute bout of exercise might also influence solid tumor physiology directly via redistribution of cardiac output. Specifically, in response to local vasodilatory signals, blood flow to active skeletal muscles markedly increases, hyperemia, to meet the increase in metabolic demand. Consequently, blood flow is redistributed away from the majority of visceral organs and inactive muscles via vasoconstriction in these vascular beds. This raises the intriguing question of how this response could alter tumor blood flow. In a mouse model of prostate cancer, tumor blood perfusion increased by operator 200% during exercise compared with basal conditions, resulting in a operator 50% reduction in tumor hypoxia, postulated to result from diminished tumor-specific alpha-adrenergic receptor-mediated vasoconstriction with a concurrent loss of myogenic tone at higher intraluminal pressures. These data demonstrate that in addition to the modulation of host factors in the systemic milieu, indirect mechanisms, exercise also modulates the TME via direct mechanisms, Fig. 3. These initial studies raise many further questions. First, given its stimulating effects on angiogenesis and blood flow in primary tumors, exercise could also impact metastatic seeding and/or colonization in secondary sites. To facilitate seeding efficiency, the primary tumor communicates with distant tissues by releasing a broad array of factors to establish a pre-metastatic niche. Primary tumor hypoxia increases the expression of the canonical HIF target gene LOX, which encodes lysyl oxidase, as well as inflammatory mediators such as CCL2 or granulocyte colony stimulating factor, GCSF. Collectively, these factors promote the recruitment of pro-tumorigenic, immunosuppressive myeloid cells to secondary sites to establish a pre-metastatic niche. Hence, exercise-induced attenuation of primary tumor hypoxia could reduce communication between the primary tumor and distant metastatic sites. Second, primary tumors stimulate the release and mobilization of bone marrow-derived cells, BMDCs, which are critical regulators of primary tumor angiogenesis, promote pre-metastatic niche formation and contribute to the repopulation of tumor cells to collectively facilitate invasion and metastasis. Clinical data in individuals with and without cancer demonstrate that chronic exercise increases the number of circulating BMDCs, suggesting that, at least in theory, exercise could facilitate the acquisition of an invasive tumor phenotype or augment metastatic seeding and/or colonization. By contrast, the tortuous and poorly perfused nature of the TME vasculature creates a substantial physical barrier for the infiltration of cytotoxic immune cells and the delivery, and efficacy, of systemic therapeutic agents. In addition, certain cell types, such as cytotoxic T cells, avoid hypoxic regions of the TME. In a mouse syngeneic pulmonary fibrosarcoma model, MC205 cell line levels of tumor hypoxia via hyperoxic conditioning, with 60% oxygen, reduced adenosinergic immunosuppression via increased production of pro-inflammatory cytokines, for example, IL-2, IL-12 and CC-chemokine ligand 10, CXCL10, enhanced the infiltration and function of CD8 cytotoxic T cells and natural killer, NK, cells, reduced the immunosuppressive function of forkhead box P3, FOXP3, T cells and improved long-term survival. Depletion of T cells or NK cells completely reversed this phenotype, revealing a critical relationship between oxygen tension and immunity in cancer. Exercise-mediated normalization of the TME vasculature might therefore improve immune infiltration. Indeed, the concept that exercise-induced alteration of the TME can improve therapeutic efficacy is supported by at least 2 preclinical studies showing that the combination of exercise and chemotherapy leads to superior tumor control compared with chemotherapy alone in mouse models of breast and pancreatic cancer. Reconciliation of the potential pro-tumorigenic and anti-tumorigenic effects of exercise-induced tumor vascular normalization, and the context that determines these effects, are crucial. Areas of investigation in exercise oncology. Cancer cells must circumvent host immunity to survive and propagate. Acute and chronic exercise alters the quantity and function of immune cell types that comprise the innate and adaptive immune system in both the circulation and within certain tissue compartments, for example, adipose tissue, bone marrow and lung. In this section, we discuss the available evidence for exercise-dependent modulation of innate and adaptive immune cell types in healthy states and in cancer. Innate immunity. In healthy physiological states, acute exercise increases the number of circulating innate immune subtypes, for example, monocytes, neutrophils and NK cells, as a consequence of hemodynamic shear stress and catecholamine-induced beta-2 adrenergic receptor beta-2AR signaling. However, chronic exercise lowers the number of circulating pro-inflammatory CD1-4-CD16 monocytes and reduces the myeloid inflammatory response, as measured by reduced lipopolysaccharide (LPS), induced production of pro-inflammatory cytokines, for example, TNF, in monocytes and a diminished neutrophil oxidative burst. The anti-inflammatory effects of chronic exercise in myeloid cells are in contrast to those observed in NK cells. For example, in elderly individuals, 65 plus or minus 0.8 years, chronic exercise exposure increased the cytolytic activity of unstimulated NK cells in peripheral blood mononuclear cells against K562 cells in vitro. Exercise might also modulate innate immune cell activity in specific tissues. In healthy mice exposed to chronic exercise, mRNA expression analysis in adipose tissue indicated a shift in macrophage phenotype from a pro-inflammatory M1 state to an anti-inflammatory M2 state, as indicated by decreased expression of TNFA and ADGRE1, commonly known as F4/80, and increased expression of ICAM1, which encodes intercellular cell adhesion molecule 1, ICAM1, CD11C, also known as ITGAX, and CD163, Ref. 121. Conversely, multiple preclinical mouse studies demonstrate that chronic exercise increases the LPS-induced release of pro-inflammatory factors such as interferon gamma, IFN gamma, TNF and Illinois-1 beta from peritoneal macrophages in vitro, suggesting a shift to an M1 phenotype. Exercise might regulate innate immunity in cancer either through reprogramming or altered abundance of myeloid cells in the TME. In various mouse models of cancer, exercise promotes a pro-inflammatory M1 phenotype in peritoneal macrophages, which are generally considered to be anti-tumorigenic. For instance, in a chemically induced mouse model of breast cancer, exercise delayed tumor development and promoted M1 activation of isolated peritoneal macrophages, indicated by increased LPS-induced release of IFN gamma, TNF and IL-12, whereas peritoneal macrophages from mice not exposed to exercise exhibited a pronounced pro-tumorigenic M2 phenotype, evidenced by increased LPS-induced release of IL-10 and TGF-beta. Similarly, exercise inhibited the metastatic outgrowth of syngeneic B16 melanoma tumors in mice, and isolated peritoneal macrophages displayed increased cytotoxicity against B16 cells in vitro. It is also important to emphasize that macrophages, including tumor-associated macrophages, TAMs, have a spectrum of activation states that represent a much broader range of functional phenotypes than the over-simplistic M1, M2 linear paradigm. For example, in tumors, further subclassifications of TAM populations, for example, M2A to M2C, have been discovered, and these different TAM populations might have differential effects on tumor growth. Areas of future investigation include how exercise shifts TAM phenotypes. Basic evidence generally suggests a shift to an M1 state with exercise but also to an M2 state depending on tissue context. Whether this differs as a function of the emerging TAM-M2 subgroup classifications and whether such a shift results in either pro-tumorigenic or anti-tumorigenic effects. In terms of myeloid abundance, exercise might limit the accumulation of macrophages and neutrophils in the TME. For example, 6 weeks of forced swimming exercise delayed the growth of syngeneic Ehrlich tumors in mice, which occurred in conjunction with marked decreases in intratumoral macrophage and neutrophil accumulation. Similarly, 2 weeks of treadmill running in mice decreased macrophage and neutrophil content in syngeneic EL4 lymphoma tumors. Finally, in the APC mouse model of intestinal cancer, forced treadmill exercise reduced large polyp burden and reduced the intestinal mRNA expression of ADGRE1, F4/80, a canonical macrophage marker. Finally, the quantity and function of NK cells in cancer might be responsive to exercise. In postmenopausal women with early-stage breast cancer, 15 weeks of moderate-intensity aerobic exercise training enhanced the ex vivo cytotoxic capacity of NK cells in peripheral blood mononuclear cells. Similarly, in the syngeneic B16 melanoma mouse model, voluntary wheel running enhanced cytotoxic NK cell recruitment, which infiltrated primary tumors via release of adrenaline and IL-6, Ref. 133. Blockade of either beta-adrenergic or IL-6 signaling prevented NK cell infiltration and abrogated the antitumorigenic effect of exercise. These data indicate that exercise might alter a potential novel link between factors secreted by skeletal muscle, such as IL-6, and the tumor infiltration and cytotoxicity of NK cells. Adaptive immunity. The number of circulating adaptive immune cells rapidly decreases following a bout of acute exercise, likely reflecting the preferential movement of these cell types from the circulation into peripheral tissues. In humans, lymphocytes that enter and exit the circulation following exercise express high levels of adhesion molecules and markers of lymphocyte activation, such as ICAM1, also known as CD54, integrin beta 2, also known as CD18, and CD53, Ref. 134. Moreover, although evidence is limited, lymphocyte translocation seems to occur towards both lymphoid, for example, bone marrow, and non-lymphoid, for example, lungs, organs, at least in animal models. Acute exercise therefore appears to prime effector cells, such as T cells, to transmigrate into peripheral tissues. Chronic exercise also alters the function of circulating lymphocytes beyond the activation or expression of adhesion molecules. In elderly individuals, chronic exercise was associated with increased telomere length and reduced commitment to apoptosis in T cells compared with individuals not reporting regular exercise exposure. In related work, 6 months of chronic exercise increased the whole blood lymphocyte proliferative response to mitogen stimulation in vitro compared with a non-exercise control group. Conversely, a considerable body of literature indicates that exposure to chronic high-intensity exercise without sufficient periods of rest and recovery, overtraining, impairs adaptive immune responses and immune surveillance, leading to increased susceptibility to opportunistic infections such as upper respiratory tract illnesses. In mice, 6 weeks of high-intensity exercise, but not moderate intensity, treadmill endurance exercise, increased the proportion of CD4-CD25 T cells and upregulated FOXP3, which encodes FOXP3, expression in the spleen, indicating that higher intensity training might elicit a T cell-mediated dampening of the adaptive immune response compared with moderate intensity training. Little is known regarding the effects of exercise on adaptive immunity in cancer. In one of the few available studies, 12 weeks of moderate to high-intensity exercise in patients with solid tumors reduced circulating concentrations of select cytokines, for example, hepatocyte growth factor, HGF, Illinois 4, MIP1 beta, also known as CCL4, VEGF and TNF, in conjunction with a trend towards an increased number of activated CD4 helper T cells and CD8 cytotoxic T cells. In related preclinical work, voluntary wheel running increased levels of CD3 T cells in mice bearing subcutaneous syngeneic B16F10 melanoma tumors. However, this increase was not observed in lung metastatic lesions in the same model. Given the current paucity of data, areas of future investigation should include how exercise regulates lymphocyte transmigration to peripheral tissues, particularly tissues that harbor cells primed for malignant transformation or malignant cells, and the induction of spontaneous tumor antigen-specific T cell responses, as well as how training intensity and/or volume modulates these responses. As described, evidence supporting the effects and mechanisms of exercise in the regulation of immune function in healthy states and in those with cancer is limited. Accordingly, the following TME-specific concerns are germane. First, tumor cells deplete their respective TME of glucose and produce high levels of lactate, which selectively weakens antitumorigenic cytotoxic and effector T lymphocytes. Under normal physiological conditions, this is an important process for self-tolerance. For example, recent studies demonstrated that FOXP3 expression in T cells provides a metabolic advantage within the low glucose, high lactate TME, resulting in the accumulation of immunosuppressive T cells. This raises the interesting question of whether exercise-induced reduction in nutrient and/or growth factor availability can reprogram and/or reduce T cell accumulation in the TME and/or restore the balance of antitumorigenic cytotoxic and effector T cells. Second, as hypoxia maintains T-cell abundance in the TME, the normalization of the TME vasculature through acute or chronic exercise could favorably modulate the ratios of T-cell subsets to promote an anti-tumorigenic TME milieu. Third, in addition to T-cells, macrophages are also highly sensitive to changes in lactate concentrations within the TME. Macrophages generally avoid lactate-rich regions of the TME and preferentially localize to highly perfused, oxygen-rich areas such as the peravascular niche and invasive edge. Indeed, macrophages exposed to lactate within the TME are activated towards a pro-tumorigenic anti-inflammatory M2 state characterized by upregulated expression of the canonical alternative activation markers ARG1 and MRC1. Refs 142143. Thus, vascular normalization through exercise might improve oxygenation, reduce macrophage exposure to lactate, decrease the frequency of M2 activation, or enhance M1 activation in the TME and/or alter macrophage localization within the TME. In summary, although many important avenues of inquiry remain uninvestigated, the emerging evidence reviewed here indicates that exercise can reprogram the TME to alter reciprocal communications between cancer cells and cell types in their vicinity to alter certain hallmark capabilities. In addition, we recognize that an uninvestigated question is whether exercise-dependent modulation of the systemic milieu confers uniform reprogramming of the TME in all distant tissues and organs, or if such effects are tissue and/or context-specific. Nevertheless, bone, which is the most common site of distant relapse and metastasis in a broad array of solid malignancies, is also a tissue that is highly regulated by exercise. Therefore, it might represent an exemplar of how exercise might modulate the TME in distant tissues. Box 1.